Basic Principles of Heredity, Part 2. All right, so now we have Mendel's work being discovered by the scientific community. And so many people were actually working on things to further his work, to see whether it applied to this organism or that organism. And really, initially, people were trying to see whether they could disprove his work, basically saying, well, this might be a fluke. Let's try and see if it's the same in this organism or that organism, or repeat his work. And so very early on, what happened was that those that were still in favor of blending and didn't fully believe that he had disproved blending, they latched on to something called incomplete dominance. And so what incomplete dominance is, so no, we still have this today. We're going to go over, we went over Mendelian dominance. We're going to go over incomplete dominance and codominance. But initially, what we are now calling incomplete dominance was what the people in the blending community basically looked to try and say, no, looks like Mendel's work isn't quite right. And so here's what incomplete dominance is. So incomplete dominance is basically the expression of a phenotype that is intermediate to those of the parents. So incomplete dominance is intermediate. And really what that means is intermediate. That means it's somewhere in between. And so if there is a phenotype that's somewhere in between your two parents, then it seems like it's blending, right? That sounds like, at least at first, in the initial description I've given you, it sounds like blending. And so here's an example. So these are snapdragon plants, and here's a red flower producing snapdragon plant and a white flower producing snapdragon plant. And if you were to cross them, you get pink flowers, which sounds a whole lot like blending, even looks like blending. And so this phenotype makes you think, wait a minute, no, it's blending. Blending is actually how most organisms still do their thing. It's just, these pea plants are a strange outlier. And if you leave it at that, then you could say Mendel was wrong, blending is still right. But now what I've described to you is not a highly hypothesis-driven scientific research, right? It's just basically do a cross, get something, and see what happens. Did Mendel ever stop after just one cross? No. And so we should take this a little bit further, right? But initially, what we see is that does such a phenomenon follow Mendel's laws? Well, it sure looks like phenotypically, no, it does not. But we have to actually delve a little further. So while such phenomena are complex, the genotypes in these instances actually strictly do follow Mendelian inheritance, and let me show you what I mean by that. So basically, first off, incomplete dominance is rare in humans. So there aren't too many examples of incomplete dominance in humans. But in snapdragons, however, flower color is controlled by two dominant genes, and so you get incomplete dominance. So here's the difference. Did you hear what I just said? Two dominant genes. Remember, Mendelian dominance is one gene, right? And there's going to be a dominant allele, and there's going to be a recessive allele. And so in the case of incomplete dominance, we're talking about dominant. And so we have two dominant genes, or you could even have two dominant alleles of the same gene. And then you end up, if they're both dominant, they're both being expressed. And so, I'll kind of give it away. I'll say what's actually happening here so you can kind of see it as opposed to having to figure it out for yourself. If you have basically let's say that we have an all red flower producing plant. So if we were to write that out in genotype, we would say I don't know, capital R, capital R. And then here we have an all white flower producing plant. And so let's call that, so actually the terminology in snapdragons is this is R1, R1. This is going to be R2, R2. And so it is dominant. So then if you cross those two, as I'll show you in just a moment, you're going to get some R1, R2s. And so here we go. So in snapdragons, flower color is controlled by two dominant genes. We have the R1 gene, we have the R2 gene. In the red flower producing plant, it's going to be R1, R1. Let's say it's true breeding. In the white flower producing plant, we'll say it's R2, R2. And I will do my best not to say D2 at any point. So basically, crosses between the two homozygous dominant strains, R1 and R2, are going to give you pink flowers, right? So they're going to give you an R1, R2 genotype. And if they're both dominant, they're both being expressed, that means you're making red pigment and you're making white pigment. And if you mix red and white paint, you are going to get pink paint. But that would be blending. This is not blending because you have cells expressing red pigment, you have cells expressing white pigment right next to each other. And so if you really zoomed into these flower petals, it would be actually, if we could zoom into the screen, then we would actually see that. There's pixels, right? Pixels making different colors. And then the like from far away, we see all sorts of different colors. But in the case here, imagine we have, the equivalent of pixels, right? So red pixel, white pixel, red pixel, white pixel. And then as you zoom out, it just looks like pink from afar, but it's not. There is no pink. There is no such thing as pink in this flower. It is red and white and red and white and red and white and red and white. And then when you zoom out, it appears pink. So you have the appearance of an intermediate phenotype, but it's actually not. So here's what we just talked about. Here's the F1. So the F1 are going to be R1, R2. And the reason why we know that it's actually not pink is because, as Mendel did, if you were to be scientific about this incomplete dominance and took it out another generation or 2 more generations and actually monitored your results, you would see. So here we go. So basically, let's do a Punnett square. So here's the original P1 cross. 100% of the offspring are going to be pink. That's nothing new. We've seen that before. I guess I take that back. That is kind of new because it's an intermediate phenotype. But seeing that homozygous dominant and homozygous dominant give you a heterozygous outcome is nothing new, right? We know that if this is the only, R1 is the only gamete that the female contribution can be. R2 is the only allele that the male gamete can take in terms of contribution, then you're going to get all R1, R2. But then, and if you stop there, then you can say, Mendel's done, Mendel's wrong, it's blending. But of course, if you take this just one generation further, so you could do an F1 cross. So that's going to be crossing R1, R2 times R1, or not times, R1, R2 by R1, R2. And so you could see that here. And then what you could see is, yeah, you're going to get some pink, so here they are, but you're also going to get back the all red flower producing plant, and you're going to get back the all white flower producing plant. And so there was no blending because you didn't lose the red or white flower production. It was just kind of covered up, right? I mean, it wasn't even covered up in a masking sense. It was just you got this intermediate phenotype because they were both trying to push through. They were both trying to be expressed at the same time. And so if you have red and white fighting to be seen, if they're both visible, then that ends up being pink. So you can see the F2 and the numbers were very consistent. 25% all red, 25% all white, and then another 50% of the offspring were again pink again. And then you could just keep following it out like Mendel did, and you would see that that's the genotype that works out to be. So before we get to codominance, so I told you incomplete dominance is very rare in human beings, but there's this other thing called codominance. And so now codominance is something that you do find in human beings all the time. And so before we get to that, let's talk about Mendelian inheritance, so just regular old dominance in human beings. So there are over 5,000 traits that have been found to follow Mendel's laws in humans. And albinism, I've talked about that in the past, right? Albinism is an excellent example because this is one of those few cases where you have a very binary outcome. It is either you make melanin or you don't. So all of the people of various skin tones count as one category, yes, they have melanin. And then albino individuals count as the other category. So it's very binary, very, yes or no, just like what we saw with Mendelian, with the pea plants Mendel was working with. Now, I don't remember if I've said this. If you want a definitive source on genetic information, then OMIM is way better than, say, Wikipedia, because OMIM is basically curated by Tufts University. And so it's online Mendelian inheritance in man. And so basically this would be the accession number for albinism. So you could learn everything you ever wanted to learn about the genetics of albinism in OMIM or any other genetic condition, disease, ailment, or just basically gene of interest by going to that database. But let's look at albinism. There we go. So the only way that you have an individual that's albino is if you have a homozygous recessive individual, right? So they lack the ability to make melanin because both of the genes necessary for a key component, a key enzyme in the production of melanin are just not present. And anyone carrying even just one dominant melanin allele can make enough melanin to have pigmentation in their eyes. Their eyes don't appear red because the iris is pigmented, so you don't see blood vessels in the back of the eye, which gives that red color. Pigmentation in the hair, pigmentation in the skin, pigmentation wherever there may be pigmentation. So if you had two heterozygous individuals, so just as a as an example of Mendelian inheritance in human beings, if you had two heterozygous individuals, neither one is albino, but they would both be carriers. And so let me ask you, well, it's actually up there. So I was about to ask, what's the chance of albinism per pregnancy? And so there you could see it's 25% because you could just set up a Punnett square. You could say, big A, little a, big A, little a, and then just calculate it out. So let's do that. So here we have the heterozygous individual. The gametes they can produce are going to be either big A or little a. And so we can look at a Punnett square to say, okay, well, they have a 25% chance of having homozygous dominant offspring, male or female, doesn't matter in this case. And so that means that they have a 25% chance of having a child that is no longer a carrier of albinism. There is a 2 in 4 or 50% chance of them having a carrier, and there's a 1 in 4 chance of them having an albino child, even though neither one of them are albino. And so you can see there's that 3-to-1 normal to albino ratio. That's what we would expect because we're talking about Mendelian inheritance, but now we're talking about human beings. And the reason why I'm doing this is because it's not common. Like in human beings, the phenotypes are generated by multiple genes. We'll talk about this. They're called polygenic traits. And pretty much any phenotype you can think of other than albinism, for example, like your hair color, your eye color, all sorts of different things, your biochemical, different aspects of your biochemistry, I should say. Those are all going to be polygenic, multiple genes, multiple alleles, multiple things going on. Here's actually, so we did talk about a dihybrid cross, right? And so you could, obviously this isn't, you wouldn't set this up as a cross, but there are more than one genes that are passed on using Mendelian inheritance in human beings. So here's an example of that 16 square Punnett square that we talked about earlier when we were doing smooth versus wrinkled and yellow versus green in pea plants. This would be the inheritance of two traits that both follow Mendelian inheritance in human beings. So if you were to combine them, well, okay, so we've got albinism we just talked about, and then the D stands for hereditary deafness. And so hereditary deafness is basically, as the name implies, it's something that's passed on, and then you're born being able to hear, but then over time you lose your hearing. And it's not like, like a much older person losing their hearing just because of, wearing, earbuds too long or something like that. We're talking about as you're pretty young, you're by teenage years, it depends. It's very variable for many different people, but let's say about by your teenage years you start to lose your hearing, and then by the time you're oftentimes by the time you're in your late 20s, early 30s, it's gone. And so what was it? I forgot her name, but there's an actress in the recent remakes. Well, I guess they're not remakes, but the recent movies, Creed and Creed 2, and she's one of the main characters, and I cannot remember her name, but she, in that movie, her character has hereditary deafness. I noticed that when I was watching the movie that they were talking about that, and then of course you might recall she's a musician, so it's even more important to her to maintain her hearing, but she has hereditary deafness, so it's going to go. And so basically you could see that if you had two carriers, right? So not albino, not deaf, not albino, not deaf, not in any danger of losing their hearing or anything like that. If they were to have a child, you can figure out the probabilities, right? So this doesn't mean like, if they had 16 children, that they would have one of every single one. That's not how that works. It's per pregnancy, right? There's a 25% chance that they are going to have an albino child per pregnancy, right? And that's just from the previous slide. Now we're looking at, okay, well, what's the chances of them having an albino deaf child? And so you can see that that's going to be a 1 in 16 chance, because you have a 1 in 4 chance for albino, 1 in 4 chance for deafness, and then you just multiply those two probabilities, 1 in 16 for albino and deaf. So that's how you multiply. It's the multiplication rule, and that's how you figure out the probabilities when you have multiple different independently assorting phenotypes. So what you can see is that they're both carriers, so they can have gametes that are capital A, capital D, capital A, lowercase d, lowercase a, capital D, lowercase a, lowercase d. And so, we did this before, so I won't, overdo it, but you have 9 that are going to be just fine. 9 out of 16 chance. So your probability at 9 out of 16 chance, you can just, divide that and get your percentage. And so that would be, what is that? That would be about like a little over half. So what is that? 0.5625%. If I did my math right. So forgive me, I did it in my head. But basically 0.5625%. So about 50% chance of, you can see that here, a little over 50% chance of being normal pigmentation, normal hearing, at least in the phenotype, right? Then you could also have, normal pigmentation, but hereditary deafness. You could be albino pigmentation, but normal hearing. And then, of course, we have a 1 in 16 chance. And so that would be, the least likely outcome, but still a possibility every pregnancy between these two individuals of having a child that is both albino and will be deaf. Okay, so of course, experimental crosses are not possible in humans like those in pea plants. And so what we do is we actually figure out what the genotypes are likely to be from familial patterns of inheritance. So it's called pedigree analysis. I mentioned it earlier. And you basically have to interview people and get as much medical data as you can and try and construct a family history to figure out their pattern of inheritance. And from that, you can get an idea of whether something's dominant or recessive. And then you can give people an idea, because usually they want to know what's the chances, because usually something happens and they want to know, well, if we have another child or if we try again, what's the chances of this happening? And you give them a probability. You can't tell them exact numbers, but you can at least tell them a probability. And so human genetics really begins with the study of the family history. And so pedigree construction is essential to human genetics, and it is tough. People lie, right? They do not want to share medical information, or they're just, they don't lie like in a nefarious way all the time. Some do, right? But others, they just basically not sure, right? Like, so if you interview me, or I have an older sister, if you were to interview her and ask about my family history, and I'm just making up a hypothetical here. I do have an older sister, but that's not part of the hypothetical. But if you were to ask us, like, oh, okay, so your grandfather's brother, did he have blah, 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 right? I don't have a lot of information about my grandfather's brother. My grandfather passed away when I was, I don't know, maybe like 12 or 13. And he was in a different country, right? Like I was born and raised here in the United States. He was, lived his entire life in India. And so I may recall hearing about certain things and say, oh yeah, no, I'm pretty sure I heard that blah, blah, blah. And then my sister could very well, meaning well, say, yeah, no, actually, I think that was his other brother that was like that. And so you, as somebody trying to construct you as a genetic counselor, which would be the profession that tries to construct these pedigrees and then use them to counsel individuals about their medical history or their future prospects, you would have to basically do the best you can with what information you have. And so, you try and gather as much information as you get, and then you'd make your best guess in terms of what the pattern of inheritance is and what a family history is, and that would be how you construct your pedigree. So here's why I'm bringing all that up right now. There is a section in this chapter that I would ask you, please do not breeze over. It is a little out of place because we'll get into pedigrees and everything in the next chapter. But for some reason in chapter 3, they have all the information about how to read a pedigree, but then we actually go over pedigrees in the next chapter. It's just this little blurb that's in there that you would think it's section 3.5. You would think that it doesn't belong there because it doesn't. But please don't just skip over it. Make sure you do pay attention to that and refer back to it when we get into pedigree construction so that you can actually have the basics of pedigree construction. So that's, section 3.5, specifically the section on pedigree construction and the symbols we use. That's really important. So don't breeze over that. Now, getting back to what we were talking about, let's get back to what I mentioned was the third form of inheritance so far that we have discussed. So there's Mendelian dominance, which is straight up dominance. It's basically you have like one gene and you could have a dominant allele and a recessive allele. And so the dominant allele masks the recessive allele when heterozygous and you get the dominant phenotype. Or in order to get the recessive phenotype, you have to get homozygous recessive because it can't show without the dominant allele not being present, right? Then in incomplete dominance, we have two or more dominant alleles, and they're both being expressed. They're both trying to show through in a phenotype, and because of that, they compete with one another and give you an intermediate phenotype. Now we have codominance. Codominance is two or more dominant alleles giving you two or more, right, dominant phenotypes. You see both things. So in codominance, full expression of both alleles is seen in the heterozygotes. So you can imagine in that heterozygote we had before with snapdragons, if it was R1, R2, R1 coding red flowers and R2 coding white flowers, incomplete dominance tells us we're going to see pink flowers in that plant. What is codominance going to tell us? It's going to tell us that we are going to see the R1 giving us red flowers and the R2 giving us white flowers on the same plant. You see both phenotypes on one plant. And that's what it means. Both alleles are seen in heterozygotes. I mean, both alleles are going to come through into the phenotype. So it also follows Mendelian inheritance. The genotype is, everything's following Mendelian inheritance, but the phenotype is not intermediate, and it's not one or the other, the dominant versus the recessive. It's both of them. And so it's not the same as incomplete dominance because unlike red and not red giving pink, codominance with those snapdragon plants would be when two different alleles are fully expressed. And so incomplete dominance is an intermediate phenotype, right, pink, whereas codominance is going to be basically red and white flowers on the same plant. So both phenotypes. So hopefully you see the difference there, right? So let's do a quick recap because I need you to know the difference between dominance, incomplete dominance, and codominance. And sometimes dominance is called Mendelian dominance. So dominance is one phenotype dominated or dictated by the dominant allele. The recessive allele doesn't get to play. Whereas if you had so these are all heterozygous, all right? So we have, let's say, big S, little s. So we get a smooth seed producing plant, no wrinkled, okay? That's Mendelian dominance. Incomplete dominance would be, okay, we have a heterozygote again. We have R1, R2. So we have the genes necessary to code for red flowers and for white flowers, or at least the pigmentation that gives us red flowers and white flowers. And then in incomplete dominance, I can't say this enough. Your clue is they both start with N. Incomplete dominance gives you an intermediate phenotype. And so what that means is you're going to get pink flowers, which really looks like blending, but we know it's actually not because the alleles don't get blended and lost forever. They reappear in the F2s. All right, so then we have codominance. Codominance is if you had an R1, R2 in our hypothetical, snapdragons are going to go incomplete dominance. But if you had R1, R2, you have a snapdragon plant with both red and white flowers separately on the same plant. So you're seeing both dominant alleles being fully expressed. And so that's why it's right here, right? So codominance is when both dominant alleles are fully expressed. Now, there are many genes, especially in human beings, there are many genes that have more than two alleles. So, you know, like pea plants, we were limited to just two alleles. That was a very lucky thing for Mendel because it reduced the number of variables he had. But for us, there are lots of genes that have so many alleles. We don't even know how many alleles are out there for certain genes. And so, but we, human beings, are diploid. So that means that we have two chromosomes. We have two, I should say, let me back up a second. We have a pair of each of our chromosomes. So that's two chromosomes. So that means that there are two loci. So that means that we carry a maximum of two alleles for a particular gene. There may be 20 alleles out there in, you know, let's go with, actually here, let's just say 5 alleles out there in the gene pool in all of human beings all over the planet. But you, let's say the alleles, I don't know, A, B, C, D, E, right? But you carry only two places to carry those alleles or have those alleles, those two loci, one on each chromosome. And so therefore you could have any of those, any combination, right? So you could have, A and B, A and C, A and D, A and E, and then I could just keep going, right? So B and, oh, I'm sorry, I didn't even do A and A, right? You could do, B and B, bed and breakfast, I guess. But B and C, B and D, B and E, etc. It just keeps going, right? So you're just going to have two. And then the thing is, though, if there's codominance, you'll express both. So the ABO blood group system is an excellent example of codominance. So our blood type is determined, well, our ABO blood type is determined by, so this has nothing to do with, plus or minus a rhesus factor. Our ABO blood group is, or our ABO blood type is, so these are biconcave discs, right? These are erythrocytes, red blood cells. And there are little oligosaccharides. So little, there's a glycoprotein with like basically some sugars, an oligosaccharide. And then the tail sugar, the last sugar, is the one that is the antigen that determines what blood type we are. And so these are all encoded by one gene. So this is the immunogenicity gene, so the I gene. And we have two alleles, right? Because we are diploid, so we have two loci. So we could be like this individual, right? Let's say if you're just looking at the cell alone, you would say, okay, well, they're making only A antigen, so they are probably IAIA. They basically carry two copies of the A allele of the immunogenicity gene, right? And so that would mean they're type A. But turns out that the three alleles that are out there in the gene pool are IA, IB, and IO. And IO is one where you lack an antigen at the very, like the last sugar is just not there. So you're not type A, you're not type B, you're not any type. You don't have an antigen, so you're type O. So this individual could very well be IAIO. Right, and so a better way to depict this on a slide would be actually if I had added a couple extra just blank lines, right? If I had blank lines, then that would be the IO one. So you'd have a little bit of sugar, but not the last sugar, which is the antigen. And so that works for all of these, but the easiest one to see codominance, all of these are showing codominance, but the easiest one to see codominance is if you're type AB. Right, if you're type AB, then you have the IAIB genotype, nothing else. And so you're expressing A antigen and B antigen. And so you're type AB. You are actually, you should, feel free to be a daredevil and do all sorts of stupid things that, you might get injured and lose some blood, but who cares? Because you can get a transfusion of any type of blood, right? If somebody transfused type A blood in you, you're good. You have A antigens. You're not going to recognize that as foreign. If you got type B blood, you're good. You're not going to recognize that as foreign. You have type B. If you get AB, you're good. You're the universal acceptor, basically. If you get type O blood, there's no antigens. Anybody can take type O blood. And so you're good. Whereas if you're type O, if you've ever donated blood and you are type O, and you have to give them your information, right? So you probably know where I'm going with this. So I am not type O. I am B positive, but what's unfortunate, I guess it's not unfortunate, but one time I was just sitting there donating blood, right? And so I'm just sitting there bleeding. And, somebody walked in. I was on that big, I can't remember, what is it? Is it Vigilant or it's the big bus that like comes to campus every now and then and says, you're a hero on the side. But I was donating blood and, and they were nice enough to me, and I donate a lot of blood, right? So this is a long time ago. I'm a fairly old individual. And so this is back when they had little punch cards and you could like actually win things by donating a certain number of times, that sort of thing. And so, I had actually completed my little punch card and I had the opportunity to get a little plush blood drop, like this little like stuffed animal, but it was a blood drop. And most people would think it's cute. I thought that was just weird and creepy. So I was like, no, thank you. And also I have two like twin girls and they were very little at the time. And so if I brought just home one of those plush little blood drops, that would be like toddler death match, right? So I just didn't want to deal with that. So I refused it and I'm just sitting there bleeding. And then somebody comes up and they come up onto the bus and they are type O. And the demeanor of the phlebotomist was amazing how she changed. She was very nice to me. She was just pleasant and polite. But when the person came, she's looking at the little clipboard thing that they filled out and she's like, oh, you're type O. right? And she was so excited and just talking how wonderful it is that they were there. I mean, they got the gold standard treatment. She offered the person who didn't have a punch card a little plush blood drop, and I'm like, oh, that makes sense. I refused it. It's, you know, so, and then she's like, and then that person said, oh no, but I have two kids, and so she got her another one, and I was just like, oh, what the hell, man? What am I, chopped liver? So if you're type O, then you know that you are the universal donor. The reason why you get such special treatment, and the reason why you also probably get a hell of a lot more emails and requests to come donate blood, is because you don't have the antigen. This is still codominance. This is the fact that you're expressing two, you know, if I drew just little empty lines, right, you're expressing oligosaccharides that lack the sugar at the very end. And so you are going to have a lack of antigen, and that lack of antigen is going to be what makes you the universal donor. So your blood will not be rejected, at least in terms of antigenicity. It's not going to be rejected by a type A person. It's not going to be rejected by a type B person, or of course type O person. The other thing they tell you is that, of course, this is for your own good, because the only type of blood you can accept in transfusion would be a type O blood. Now, this is becoming a little less relevant because just earlier this year, there was actually, I guess it was 2019. There is a study that was published, and it's just the first one, but there was a technique that was published that seems very promising and very likely to be an actual technique that will be used extensively in the future, that can strip all the antigens off of blood cells. And so it can turn anybody's blood into type O blood. And so, haha, your days are numbered as the gold standard of blood donation. But I will say this, and this is scientific data that I've gathered. Type AB individuals are universal acceptors, right? And type O individuals are the universal donors, and they seem to get more cookies when they donate blood. They, you know, literally the woman said, here, take as many as you'd like to the type O individual when they're done. Whereas mine was just like, okay, make sure you grab a cookie, right? And so I just noticed the way she really emphasized the A cookie, right? And so, you know, I took two anyway because I was upset. So that concludes this section. What we'll do next is we'll get into chapter 4. So we'll start to talk about some modifications to these Mendelian ratios and all the other things that people were able to learn as we increased our knowledge of genetics after Mendel's work was rediscovered.